Okay, picture this. You're a detective at a microscopic crime scene. Your only suspect? An unknown bacterium. Totally invisible. So, how do you unmask it? Today, we are cracking the code on how scientists use DNA to identify these tiny mystery microbes. All right, let's dive in.
And that's the big question, right? You can't use an eyewitness sketch or a lineup. The identity is hidden deep, deep inside its genetic code. To solve this, we have to find its unique, unchangeable signature. What is basically its genetic fingerprint.
So, our investigation kicks off with a hunt for the perfect piece of evidence. We're looking for a clue that exists in pretty much all bacteria, but is still unique enough to tell one species from another. Well, it turns out that perfect piece of evidence is a gene called the 16S rRNA gene. You can think of it as a universal barcode for bacteria. See, every bacterium has this gene, but the exact sequence of its DNA letters, the A's, T's, C's, and G's, acts as a totally unique fingerprint. It lets us pinpoint its identity. Okay, so we have our target, that 16S gene, but how do we actually read the fingerprint? For that, we need a special kind of forensic technique to lift that print right off the DNA evidence itself.
So this technique is called dideoxy sequencing, or you might hear it called the Sanger method. And the core idea? It's actually brilliantly simple. We make thousands and thousands of copies of that fingerprint gene. But, and here's the trick, we use special ingredients that randomly stop the copying process at every single letter along the way.
Okay, so let's break down how this actually works. First, you start copying the DNA, but then you add in these special terminator bases, and each one is tagged with a unique color. These terminators randomly stop the process, which creates this whole collection of DNA fragments of every single possible length. Finally, a laser sorts all these fragments from shortest to longest and reads the color of the very last base on each one. That sequence of colors, that's our DNA fingerprint.
So what you get at the end isn't just, a boring string of letters. It's a visual map called a chromatogram. Each one of these colored peaks you see represents a single base in our DNA sequence. This right here is our raw evidence, fresh from the lab. Now, just like any evidence from a crime scene, not all chromatograms are created equal. So our first job as the investigators here is to figure out if we have a clean clue or if our evidence has been, contaminated.
Okay. This is what you're hoping for. A high-quality chromatogram has these sharp, individual peaks. Each color is totally distinct, and there's no confusion about what base is at each position. It's like getting a perfect, crystal-clear fingerprint. It's easy to read and super reliable.
But then, sometimes you get this. It's just a jumbled mess of overlapping peaks. This usually means your sample was dirty. Maybe it had DNA from more than one bacterium, or there was a problem with the chemistry. This is like a smudged fingerprint. It's basically useless, and you've got to go back and start all over again.
And here's the thing you have to remember. Even in a really good sequence, the very beginning and the very end can be a little messy. As the investigator, your job is to find that sweet spot in the middle, the really clean, high-quality section, select it, and copy it. That's the part of the fingerprint we can actually trust. All right, so once we've got our clean DNA fingerprint, it's time to identify our suspect. And to do that, we have to run our sequence against a massive international database of all known microbes.
We access that database using a tool called BLAST. Seriously, think of it like the FBI's master fingerprint database, but for DNA. It's this incredibly powerful search engine that takes our sequence and compares it against millions of known sequences that scientists from all over the world have submitted. And you know what? Using it is actually pretty simple. First, you pick the right program for DNA, which is nucleotide BLAST. Then you just paste your clean, high-quality sequence into the search box. Now, here's a key step. You tell it to search specifically against the 16S database. This really focuses the search and gets you relevant results way faster. And then, you just click BLAST. The system takes over from there.
And just like that, moments later, you've got your results. This is it, the final stage of our investigation, where we analyze the matches and officially name our microbial suspect.
So, what BLAST spits out is this ranked list of the closest matches. And hey, notice how all the top hits for our sample are from the Pseudomonas genus? That's a group of really closely related bacteria. Now, to be sure we have a match, we need to look closely at two key numbers. Okay, so the first number we need to look at is called query coverage. This just tells us what percentage of our fingerprint, the sequence we put in, actually lined up with the sequence in the database. Getting 99%? That's awesome. It means basically our whole fingerprint found a matching section to compare against.
All right, the second number is percent identity. This asks a slightly different question. Of that 99% that lined up, how much of it is a perfect match, letter for letter? In this case, it's over 98%, which is a really, really strong match.
So when you've got high scores in both of those categories, query coverage and percent identity, we can say pretty confidently that this part of the case is closed. Our mystery microbe is without a doubt a member of the Pseudomonas genus. But wait, the story isn't always that simple. Technology is powerful for sure, but it's not perfect. Sometimes the computer's report flags a potential conflict, and that's where the human detective, the scientist, has to step back in.
And this is where that teamwork between the machine and the human expert is just so important. So look, on the left, the BLAST report might say, hey, there's a mismatch here. Your sequence has a C where the database has a G. But then a scientist with a good eye can go back to that original chromatogram on the right and spot the truth. There's a tiny little black peak hiding under that big blue one that the machine's software totally missed. It's a perfect example of how the final call often comes down to expert human judgment.
Okay, so let's quickly run through our detectives toolkit one more time. The 16S gene? That's our universal fingerprint. Sanger sequencing is the technique we use to lift the print. The chromatogram is the raw evidence we have to inspect carefully. BLAST is our massive database of suspects, and in the end, it's your scientific judgment that puts all the clues together and cracks the case.
So think about what we just did. We've taken one single, invisible microbe from a sea of possibilities and given it a name. This exact same process is what helps doctors diagnose diseases and lets scientists monitor entire ecosystems. It's happening in labs all over the world every single day. It really makes you wonder, right? With millions of species still out there, completely undiscovered, what secrets will the next genetic fingerprint reveal?